Welcome everyone. You've landed at the CyVerse Focus Forum webinar series. I'm Tina Lee, CyVerse User Engagement Officer, and today's presentation is by David LeBauer on getting grounded in TERRA REF, a publicly accessible high-res sensor database for crop phenomics. David is the Director of Data Science for the Agricultural Experiment Station here at the University of Arizona and the Data and Computation Lead for the TERRA REF project. He is, and not just for today's webinar, outstanding in his field, as you can see. For those unfamiliar with CyVerse, we're an NSF-funded cyber infrastructure project. Our webinar series is designed to fulfill part of our mission to train scientists like you on how to use CyVerse's computational resources. Let's quickly take care of some housekeeping, and then we'll start the webinar. David's presentation is roughly 30 minutes with time for Q&A at the end. Please open the Zoom chat window. David will answer questions there at the end of his webinar. Later on today, we'll post his slides and the video recording on our website page for you to review at any time. CyVerse just concluded our third successful container camp, and our 22 participants are now off containerizing to their heart's content. We're also working on tweaking the curricula for both container camp and our other training, foundational open science skills, to accommodate remote learning. So watch our website for more information on these trainings. Without further ado, I'm pleased to introduce David LeBauer. Hi, David.
Hi. Thank you for the introduction, Tina.
You're welcome.
And thanks to everybody else who's able to join this talk and or watch on the recording. And so today I'm going to tell you about this project, TERRA REF, that I and many other people have been working on over the course of the past about 5 years. And we're very excited at this point to be right on the cusp of releasing our data set to the public. So I'm going to give a brief introduction to high throughput phenomics and then talk about why we have invested so much in a reference data set. I'll describe this system that you see over on the right of your screen, the field scanner, and the sensors it has and the phenotype data that it generates. Toward the end, or actually I'll spend quite a bit of time talking about how to access the data. And finally, I'll conclude with the open phenomics pipelines that we've been developing and moving the community in that direction. So both CyVerse and I and many of us are interested in open science, and I just wanted to briefly mention that agriculture and open research have a really long and successful history. In fact, the reason that there are so many people in the world today has much to do with the science of breeding better crops, and people led by Norman Borlaug, most famously, who have created higher yielding plants. Not only have they created it, but they've distributed it for free. So you can see all of these seeds that have been collected at CIMMYT in their germplasm bank. Anybody's allowed to write to them and ask for some seeds in order to do their own breeding programs or generate their own seeds for larger scale production. I see a clear analogy to the way that we can share our software on GitHub and data through other means between this mechanism of using the seeds to transmit knowledge encapsulated by the process of breeding and selecting better plants. We can also do similar stuff, but even much easier, because a lot of the data and software that we have, you can just go and find and download, clone, and even contribute to. So I'm going to start out with a description of high throughput phenotyping. For some of the challenges, I apologize that my, okay, I'll just go ahead and I'll go through the entire slide at once. So a phenotype is a plant trait, pretty much anything that you can measure on an organism. Of course, today we'll be talking about plant traits. I have this image on the right, which is a corn plant. And typically this is what people like Norman or other breeders even today do, is they'll go out and they'll measure the height of their plants, etc., as they grow or at the end of the season. And the most important thing that they measure is the yield of the grain at the end of the season. What high throughput phenotyping seeks to do is increase the frequency and resolution of these measurements as well as enable a greater variety of measurements. So more specifically, look at the next picture is a 3D point cloud of a sorghum plant. And what you can see is that it's easy to get accurate measurements, not only of the height of the plant, but also of other components, the volume of the seed head, for example. So what we want to do is make it easier for people to routinely use these types of devices in order to make better inference about basic biology, as well as quicker breeding of new crops. The challenges are that the sensors are expensive and the data are difficult to interpret. So ARPA-E, the TERRA program, was a major investment by the Department of Energy to advance high throughput phenotyping. Finally, this slide on the bottom, you can see, in the blue are hand measurements and in the red are manual measurements. And you can get a sense of the fact that it takes an entire day to measure an entire field for a person. What we can scale up and do is get this nice time series. So there's a lot more that we can do as far as generating inference about the physiology of these plants. And we have high temporal resolution. So the Department of Energy set out a funding opportunity announcement. They wanted to bring together people from many different domains, from biology, plant genomics, and breeding, as well as engineering, computational biology, computer vision, in order to advance the technology and science of high throughput phenotyping. And our team was category 5. It was the reference data generating and hosting team. And so our mission was to make the highest resolution data possible available as broadly as possible. And this just gives an idea of the size and complexity of the team, the number of people who have participated in this endeavor. There'll be an acknowledgement slide at the end. So our objectives were to generate reference, sensor, phenotype, and genomics data, make it available, and at the same time, develop an open source pipeline to take the data from raw all the way to the end. And finally, to provide people with computing resources required to analyze these data sets. This machine is capable of generating terabytes a day, and so it's non-trivial to analyze data from an entire season, much less the 10 seasons that we've had so far. These are some of the sensors that are on this, the platform in our sensor box. I wanted to back up real quickly and tell you a little bit more about the field scanner system and the scale of it. So the green box that you see over here on the right is a shipping container, and the white box where the sensors live is about the size of a minivan, and it weighs a few tons. These are the instruments that are on the system. We have stereo RGB cameras. We have 2 hyperspectral cameras capturing the visible and near infrared at about 0.6 nanometers resolution, and the shortwave and infrared at about 12 nanometer wavelength resolution. We have a 3D laser scanner, gets about millimeter scale resolution 3D point clouds. The idea is to be able to get daily to weekly measurements from each of these sensors. Some, like the hyperspectral camera, take days to traverse the field, whereas others we can capture a number of times per day. We've been able to generate hundreds of traits and millions of observations per season. We also have unprecedented resolution for the genomics and the sequencing of 400 genotypes of sorghum. All of it is open. It's available now, and we're going to make a public release in 2020. So from the very beginning, we've made the data available at every stage, caveat emptor, but with people, providing people with access to it. We're trying to make it as freely available as possible. So I'm going to take a few moments to go through some examples of the imagery that we're able to collect from the machine. So this is our stereo RGB camera, taking a picture of a mature sorghum plant, and you can see the panicle or the seed head over here on the right. We can stitch that together in order to look at the entire field, but typically we analyze image by image or plot by plot. This is an image from our thermal infrared camera, where color represents temperature, and you can see the red in the background is a soil that's relatively warmer than the crop as a result of the leaves transpiring and evaporating water and evaporative cooling. These are some of the data products that come off of our 3D laser scanner. So the first thing we get is actually a flat raster file on the left, a PNG. It can then be converted into a point cloud in both PLY and LAS formats, depending, so that people can make use of them with the commercially available and publicly available GIS software. The PNG is a little bit easier to compute a lot of basic geometric properties on. This is an example of our visible and near-infrared hyperspectral data in the green wavelength. You can see this, the units on the two axes are in meters. That's relative to the field. And each pixel is about 1 millimeter in sampling distance. Finally, we have a PS2 camera that captures an image. It actually captures 101 images that capture the response of the photosystem to a pulse of light. And you can get information, including this variable FV over FM that tells scientists the efficiency of the photosynthesis apparatus in the plant. Very recently, we've been able to put together, led by Marcus Tuller and his student Ibrahim, these soil maps of 12 different size classes of soil particles, as well as the hydraulic properties, moisture content over time, organic matter, and pH. There's a lot of other data that goes along with this as well. There's the agronomic metadata that I've provided some examples of over on the right. The geospatial plot boundaries, which are key for linking cultivars or the genotypes to the images. You have information about the experimental design and the agronomic managements, which you can see in the lower right. We also have the weather data, hand measurements, a lot of derived traits, and finally the genomics data. In order to get derived traits, we'll take an image such as this. This is one example that I like to use, canopy cover, because it's simple to explain and understand. The first thing that we do is we create a mask, so we exclude the soil from this. And then basically we calculate the ratio of the plant pixels to the total number of pixels. And that is an index of a measure of canopy cover, the percentage of the area looking down at the crop that's covered by the plant. It's typically, even for a simple extractor, it's not as easy as it looks at first pass. The first thing that we do is we take the raw image and we georeference it. But as you can see, even after we have turned it into a regular geotiff image quality can vary quite a lot, whether it's shaded or sunlit and washed out, or if it's half shaded and half sunlit. This isn't like a regular point and shoot camera that automatically adjusts to the background. We have fixed settings on the camera, so we have to be able to process very diverse quality of data. In this case, we have 3 different algorithms depending on the condition of the image that comes in order to generate this plant mask and finally generate a measurement of plot level canopy cover, which you can see for all of the plants over time in the top image and as a map of the field in the bottom image. There are a number of uses of the TERRA REF data beyond crop improvement, which is kind of the core focus of the program. Even outside of biology, there's a lot of interest in the physics of the sensors, how they work in different environments, and how you can combine images or information from different sensors to make better inference. There's also a lot of work that can be done in the realm of basic biology, including the rules of life that translate genes into phenotypes and that control the interaction between genes and their environment and how that translates into what is actually observed in the field when you measure a plant. One of the earliest applications was teaching. It's been used in a number of different courses, and our tutorials are based on some of that. And finally, in the past few years, we've been using it to support a number of programs that are working on basic machine learning research. Some people were interested in the applications in machine learning research, so I just want to quickly give an idea of 3 programs that have been using our data sets. One of the earliest was DARPA's Data-Driven Discovery Program, where they had a number of different teams make it easier for scientists to use the data to make predictions about plant growth and sorghum yield. NSF funded an Ideas Lab program in order to work out gene by environment and interactions using machine learning. And finally, a large fraction of our team has been funded to do additional research into explainable AI, i.e. figuring out what and why a black box machine learning algorithm is learning from in these images. The core of my talk, I want to tell you about how to access the TERRA REF data. And I want to start with a little bit of a caveat, which is that the bulk of our effort to date has been to generate the data sets themselves. And over the past year, we've been transitioning to publishing the data and making it usable for the community. And so it's not 100% smooth. We didn't invest in a large infrastructure to make it easier to use the data itself. But now, especially with the help of CyVerse's infrastructure, we're implementing these in a more consistent environment that I will tell you about moving forward. Here's a few web interfaces that we have. One is the visualization of the phenotypes. You can find some sample data and instructions for downloading it, just to use example data sets. Most people will want to explore the types of data that we have before launching large analyses over the entire data set, and that's what those are intended for. We have documentation and finally tutorials. And to make it clear, we want to make this data as useful as possible. This represents a major investment, and part of what we were pleading to DOE, the rationale for funding it is to make it available in the public domain, not just for academic research, but also for industry and applications. So anybody can use it. Published data will be in the public domain. And other data, we have a data use and authorship policy. But even when it's in the public domain, it's going to go on Dryad. The idea is that a scientist would cite the data sources and recognize our efforts as necessary. I mean, as appropriate for the conventions of our community. The data publication is in prep, and we expect to release it almost imminently. We're going to start by releasing seasons 4 and 6, and once people learn how to use those, we'll make data available as needed or as warranted. So it's a tremendous effort to process an entire season and validate it and make sure that it's good, high quality, and useful. So as I mentioned before, because our focus hasn't been on one unified interfaces, there's a number of different interfaces that can be used to access the data, and I apologize that this is daunting. It's even daunting for me, and I've known about these throughout. But the idea is to make it available to people in as many different ways as possible. And so I'm going to go through a few ways in how I recommend a new user might enter and start learning about the data. So one is just to browse the trait data. So these are the phenotypes that we've output. And we have this website where you can look through it. We have tutorials where you can begin using R and Python to download and explore the data. And one of the reasons that R and Python are so powerful is that, and why generating a unified interface is so difficult, is because with such diverse data set, it's very difficult to predict all of the ways in which people would like to use or analyze the data. And so by making data available through APIs and in ways teaching people as an entry point to use R and Python, it provides a lot of power and flexibility that us designing an interface would be much more difficult to do. So that way we can rely on existing technologies. So this is an example of the Traitvis web application where you can browse the data. It has maps, and it also has a little bit of information about how you can download the data. A lot of this comes straight from the tutorials. We have a web interface called Clowder where you can browse the data, and it also has an API where you can access metadata about the files themselves. We generated the tutorials as a way to, as a quick start way to access the data. And as I'll explain later, there's an interface within CyVerse. You can open up an RStudio or a Python environment and begin to run the code that's in these tutorials. With data, our sample data is already publicly available. The tutorials have also been translated by Christina Reamer in my lab into webinars and videos, and we have a number of videos still upcoming. This work was funded in part by the Midwest Big Data Hub in order to make the data more accessible. So the visual and interactive computing environment by CyVerse in their discovery environment allows us to put computing environments and make them available for people. So when we tell you about the tutorials, a number of them, because of the nature of the data, require a lot of dependencies, software dependencies that may be difficult for people to install on their own computer. I say maybe, and that's generous. A number of them are very difficult to install on your own computer. Fortunately, Docker has made it easier to pre-configure these environments so that it's as simple as logging in 2 times and then pushing a button, launching your RStudio environment or your Jupyter Lab environment and running with that. So here's 2 examples of code that we've provided in VICE. Once you're ready to download a lot of large files, currently it's available. The reference datasets are hosted at the National Center for Supercomputing Applications, and we're making the data available through Globus. This, if you have permission, you can access all of it, and in a few weeks you'll be able to access seasons 4 and 6 through Globus, and then you can transfer terabytes of data to your machine. And before you download it to your own computer, some of these files are 20 or even 150 gigabytes each, just to give you an idea of the scale and size of what we're dealing with. Some of the more recent work that we've been doing in order to take the work from TERRA REF and migrate it into a more robust and flexible framework is to take the individual components, modularize them, and use a proper workflow system called MakeFlow and WorkQueue. Unlike when we developed this at NCSA at the University of Illinois, where we had a big computer with a large data set, storage available, now this will allow us to distribute the data to many different computers that are available and run it externally, and then bring the data back and ingest it into iRods and make it available through the discovery environment or an iRods client. We've also taken this opportunity to really work on making it easier for our collaborators and others to contribute new algorithms to the pipeline. During the process of the TERRA REF project, it was a big a major challenge was working with people who were developing algorithms around one or a few different select images and transferring that and being able to scale it up, not only scaling it up for high performance computing, but also generalizing it so that regardless of which season they were using or which plot layout was in the field, which crops or cultivars were there, the people developing the algorithm, trying to isolate them from having to understand all of the experimental design and really just focus on taking an array or a point cloud and converting it into a measurement of a plant. And so the templates, the goal here is to make that easier, to enable best practices, guide people gently along the way so that we can generate algorithms that will not only work on this large machine, but they can also be used for other high throughput phenotyping platforms such as tractors and drones. And so these templates, the idea here is that for a new user, the first thing that they need to do is just open one file, add a little bit of metadata in the header, and then copy and paste their new algorithm into where it says algorithm. And so this one, the template for the plot level RGB assumes that you have a rectangular image of the plot, and what can you do with it? The trivial example is just to measure the channel size and return it. But this is the template intended to be replaced with something more informative, such as the example I was using of canopy cover. So both with the TERRA REF project and now with this new, what we call the more generic ag pipeline, the idea was to have all of these components modularized, built into Docker, and able to be reused. And so we have large GitHub repositories in each individual organization, and each individual repository has one or just a small handful of individual, what we call transformers now. And it will take, like in the case of canopy cover, we'll take this RGB image and compute the percentage of the image that is covered in plants. So we're trying to make it easier for somebody who says, well, can you measure this, to say, okay, well, fork this template, fill out this file, compile it, and submit a pull request so that we can get that in and check it out and deploy it on the pipeline. It sounds very quick because I have limited time right now. The last thing I want to talk a little bit about is the fact that really what enables this is data formats, standards, and conventions. That's what enables interoperability and generality of the pipelines. And it was also a major effort within the scope of the TERRA REF project to generate data sets that either adhered to or helped to redefine new standards and conventions. So starting with the genomics data, it was actually quite easy. There was no disagreement that this was a set of data types that are standard and used throughout the community, as well as using Genesis as a reference for the identification of the accessions or the individual genotypes that we were planting in the field, or crop varieties that we were planting in the field. So that all happened very quickly and very early on. There's been a lot of work in the world of earth science and remote sensing. So there was some place to start, including the Climate and Forecasting Conventions and the Open Geospatial Consortium. They've got a number of different definitions of file types and metadata formats. And we've adopted GeoTIFFs, NetCDF files for the hyperspectral images and LAS files for the point clouds. We have a number of other data formats, as I mentioned earlier, some PNGs and PLY files, but we try to focus on these standard formats. And then with the traits, it was a lot more murky. There's ontologies based on historical methods of measuring plants, such as categorizing disease severity as a value of 1 through 5. But ontologies didn't really work immediately because there's so many more measurements that we can actually make than the ontologies contain. So in our database, BettyDB, we've just followed to some extent the way of naming things in the climate and forecasting conventions to name new traits as they come out, as we develop them. I think more importantly, and what I'll talk about in just a second, is that the Agricultural Model Intercomparison Project, in collaboration with the USDA and their ICASA vocabularies and data formats, have a standard format for writing out data that's used by agricultural models. And then there's the Breeders API, which is a specification for an API that's used to share data among breeders, among people who have breeding programs. And so none of these cover all of the cases that we need. And so we've adopted a mixture. And I'm going to show you in the next slide just a little bit about how adopting BRAPI has helped in another project that has just started about a year ago. And I'm one of about 5 groups of people with agricultural data trying to get it into the National Agricultural Library in a standard format from many different places. And so rather than just dumping out our data as we have it and writing a converter, we do have a BRAPI interface to our data, which isn't the standard that they want, but we're converting it from BRAPI into the ICASA AgMIP format, which is the one that's being led by, being used by the Agricultural Model Intercomparison Project. You'll note one of the PIs, Garrett Hugenboom, he is also a leader in AgMIP and in this project. And so once it's in that format, so that these arrows represent converters or transformers, and publishing it in different places, we'll be able to put it not only in the library, but also make it available for crop models. Once we have this conversion from BRAPI to ICASA, other databases that have BRAPI interfaces will be able to make their data available in this format as well. And that's the goal. Currently, BRAPI doesn't support all of the data that's required. So we're also using this as an opportunity to inform the BRAPI community as to how to expand their definition, their API definition, to account for a lot of the information that's going to be required for running crop models, which overlaps a lot with just the nature of what's needed for high throughput phenotyping applications. High resolution time series, more specific geospatial information about the plots, and other agronomic metadata that's currently not captured in BRAPI. So there's other groups, for example, Bruno Basso at the Kellogg Biological Station, where they have a large history of agricultural research data cataloged using the ecological metadata language, and they're working on translating data from in that format into this standard. And once they do that, similarly, other databases that are annotated with EML will be able to convert their data into this standard. This is a big picture vision where interoperability can make better research and more synthetic research possible. And with that, I'll wrap up my talk. I have to acknowledge a huge number of people who have contributed to this project. And there's many, many others that aren't currently listed here, as well as our funders, primarily ARPA-E, and to a large extent, CyVerse has provided a huge amount of support, as well as the University of Arizona. And with that, I'll conclude. We also have to thank the National Science Foundation for substantial support. Thank you.
Thank you, David. Nice detail, just explaining what it is, who can use it, how to access it. That's great, because a lot of our stuff is based on tools, but then people don't know where to find data. They may not have data. So this is a great resource for the community. Are there any questions for David? I don't see any in the chat window. So while we maybe give a minute or two for people to type in if they want, I'll briefly highlight our next webinar in 2 weeks, Friday, March 27th. CyVerse's local snake charmer, his name is Satish Perry. He's going to demonstrate SnakeMake, a workflow management tool in CyVerse VICE. You can find that in VICE. That's in response to some folks who attended the earlier webinars last season and asked for workflow management tools. So we'll be featuring Satish then in a couple weeks. And I don't see any other questions. So I think you've done a fabulous job of basically giving us all the information that people could need for accessing TERRA REF. Okay, well, thank you everybody for attending. Thank you, David. And we, as I always do, will post David's slides and a video recording of this webinar on our webinar page on our website. So thanks everyone. Hope to see you in a couple of weeks. Bye now.